Hello, my friends. Thank you so much for joining me. Today, I'm going to do another presentation on biological and biotech medicinal products. I'll be focusing the presentation on finishing off on the antibodies. I'm not sure if I'll do any more on antibodies, and then I'll just do a very short introduction on some T-cell therapies. So this presentation, I'll be talking about, giving an overview of the monoclonal antibodies, specifically talking about the treatment of cancer and tumours. The previous presentation, the focus was on the rheumatoid arthritis and Crohn's disease. The antibodies, the monoclonal antibodies, were acting as a neutralizing effect. It was neutralizing tumor necrosis factor that was implicated in the inflammation in these particular conditions. So it had a neutralizing effect mostly. However, monoclonal antibodies are used for the treatment of certain cancers and tumours, and so I'll just be giving an overview there and talk about some of the ways that those antibodies are used. Monoclonal antibodies are used in combination with other cancer treatments such as chemotherapy, radiation therapy, and certain advanced therapies, although there may be problems with combining with regards to cytotoxicity. The use of monoclonal antibodies as checkpoint inhibitors. I'll be talking about what checkpoint inhibitors are and how, the use of them to overcome this tumor microenvironment that occurs, and it means that it's not always possible to treat the tumor. The checkpoint inhibitors can help to improve those clinical outcomes. And then I'm going to do a very short introduction to T-cell therapies and just give some descriptive accounts and contrast T-cell receptor therapies to CAR T-cell therapies. Well, for cancer treatment really is what I'll be focusing on. So I just want to do a very quick recap on antibodies because it's helpful for this slide. So we know about the various regions on the antibodies. There's the antibody binding region, the Fab region, which is on the variable part of the antibody or on the complementary determining region. And it binds to the antigen's epitope, which is on the surface of the target cells, or well, it could be on the surface of, it could be externally. And then we have the FC, which is the binds to the effector cells. So it can bind to the natural killer cells, and it can bind to macrophages and neutrophils. So it binds to the effector cells. It can also bind to the complement cells as well. If it binds to effector cells, then this is an ADCC, so it's antibody-dependent cytotoxic cellular cytotoxicity. Antibody-dependent cellular cytotoxicity, and it basically causes the release of toxic molecules, cytotoxic molecules such as perforin and granzyme, and it leads to the destruction of those targeted cells or those targeted targeted cells. With regards to the FC, well, the FC, if it binds to one of the complement proteins, it can lead to membrane attack complex formation, and this is just basically the breakdown of the antigen or whatever is causing the antigen. So this is just a very top level overview. I mean, there's a lot more to it than what I've just discussed. But, we have the Fab region and we have the FC region, and these regions have very specific roles that they play. And with regards to antibodies, we can think about it as just a neutralizing effect, or it can be neutralizing anti-destroy, or to down the line. So monoclonal antibodies are used for cancer treatment as checkpoint inhibitors, and again, as I've said in the previous slide, they can be combined with other cancer treatment to, I would say, to improve the clinical outcomes for the patient. So this is just a reminder, and it's just a pictorial representation of an antibody. So we have the variable regions, and we have the constant regions, or the crystallizable regions. The constant region is where we have the Fc fragment, the various domains, and the glycan regions where we have the sugar residues, and this impacts on the activity of the antibody, and can also impact in terms of the therapeutic safety of the antibodies. We also have the hinge, which is also responsible with regards to activity and reactivity. And well, what I mean by reactivity, I mean is that it's high in terms of its binding affinity. And the disulfide links, which can shift and cause breakage, and then you can get deformation of the antibody. So we have the heavy and the light chain, we have the antigen binding sites, and we have the disulfide links, we have the hinges, we have the glycan regions, and we have the Fc region, which is responsible. The Fc region binds to the effector cells, and then that causes the antibody dependent cellular cytotoxicity. Right, so checkpoint inhibitors. So a checkpoint inhibitor is a type of immunotherapy, and what it does is it blocks inhibitory signals on immune cells. So inhibitory signals, they're naturally occurring. They occur because we don't want to have this overactive, overstimulated immune cells that will kill infected and cancerous cells, but may also start to attack healthy cells. So the body has a way of controlling and regulating that, and ensuring that that overexpression, that overactivity doesn't occur, and we don't get autoimmune diseases. So that's the purpose of checkpoint inhibitors. It's to neutralize the effect of the the purpose of inhibitors and inhibitory signals is to dampen and to control the and regulate, if you like, the T cells so that they're not overly active. And the purpose of checkpoint inhibitors is to neutralize that effect so that we don't get that dampening on, and we do get more activity because of the effects of the tumor microenvironment. The tumor microenvironment takes advantage of the body's natural inhibitory signals by either overexpressing the molecules that are responsible for it, or other things that I can discuss in the later slides. And what happens is you create a tumor microenvironment where it's very difficult for the T cells to actually get to the tumor cells to destroy them. And so these checkpoint inhibitors, they kind of neutralize that microenvironment, allowing the T cells to actually get to the tumor cells and to actually destroy them. The checkpoint inhibitors that I'm going to be talking about are listed here. Some of them are, not all of them here. So we've got PD-1 and PD-L1. So these PD-1 and PD-L1, they're only expressed, well, PD-1 is only expressed on activated cytotoxic CD8 T cells, and they're the programmed death cell proteins, and they have an inhibitory receptor. As I said, they're only found on the activated T cells, but when they bind to the PD-L1 I've got here, but there's also PD-L2, which are found on cancer cells, they inhibit the signals, and they inhibit the T cells, and they down regulates, if you like, the T cell activity. And so what's happened is that the tumor cells overexpress the PD-L1, so that the chances of of binding with the PD-1 is increased, if you like, and therefore they're more likely to evade the immune system. And this is how well, partly how the tumor microenvironment is formed. Partly, it's not just that. CTLA4, that's cytotoxic T lymphocyte associated protein 4, and what happens is that there's another receptor, which is the CD28 receptor, which binds to B7 molecules on the antigen presenting cells. So the antigen presenting cells are like dendritic cells, and they have to get this CD28 binding to the B7 molecule, and it's this co-stimulatory requirement. So for T cells to be fully activated, they need to have that co-stimulatory requirement. But the body has a way of you don't want to have too much activation. As I've said, you want to have a controlled system so that you're not attacking, or that the body's not attacking its own healthy cells. So they have this, the body has this CTLA4, this cytotoxic T lymphocyte associated protein. This is, as I say, it's normal T cell immune function, and it's to reduce overactivity. But the problem is that tumor cells exploit this process. Obviously, it happens, and the tumor cells will exploit it. And then we've got this LAG3, lymphocyte activation gene 3. So this is found on exhausted T cells, and it reduces their functions. And some cancers increase this LAG3 expression to make the T cells less effective. And so there are LAG3 inhibitors, mAb, sorry about that are being studied. They're being studied for to address this. But that's a way that tumor microenvironments are formed. And then we've got this TIGIT, which is a T cell immunoreceptor with Ig and ITIM domains. Now this TIGIT competes with another surface molecule or surface marker called CD226 for the binding of something, a CD155. So this TIGIT is like an off switch for T cells and for natural killer cells, and tumor cells, they take advantage of this by increasing the CD155 expression. So CD155 binds with CD226, but if the CD155 comes across this TIGIT, then it will decrease it's like an off switch, so it'll switch it off, and it'll switch off the T cell expression. So the tumors can take advantage of this by increasing their CD155 expression, and therefore again avoiding T cell and natural killer cell destruction. Another thing that tumors do is they reduce the major histocompatibility complex class 1. They reduce that so it's not so many, and so you can't get that expression of the mutated cells, so that the T cells can then attach to the peptides, and then instigate that destruction of those cells. So that's another thing that tumor microenvironments do. They reduce the numbers of major histocompatibility class 1 on the surface of their cells. So I just want to talk about just 2, I guess, checkpoint inhibitors. So I did talk about PD1 and PDL1 in the previous slide, and I said that PD1 is expressed on activated T cells and PDL1 is expressed on the tumor cells respectively. And I did say that this PD1 binds to the ligand PDL1, and it suppresses this T cell activation, and it's just a way of the body preventing autoimmunity. But PDL1 is one of the molecules that are overexpressed on tumor cancer cells, and so therefore when you get this binding, because it's overexpressed, the T cells become less effective at killing the tumor cells. The T cells' ability to produce all the important cytokines is reduced, and the proliferation in order to then go off and kill more cells is decreased, and the ability to respond to tumor antigens is decreased. And so that's the reasons why you get this formation of this microenvironment. One of the reasons, it's not just that. As I said, it's also the reduction in the major histocompatibility class 1 molecules that are presented on the surface, as well as other things. So that's one of the reasons. And you know, monoclonal antibodies that bind to PD1, so they bind to the T cell receptors that prevent that from happening, and PDL1, which is on the mutated cells, that blocks that binding interaction, and therefore restores the T cell activity, and it then enhances the T cell's ability to attack cancer cells. I just want to talk about another one, another checkpoint inhibitor, and that's the anti-CTLA4. CD28, first of all, is a co-stimulatory receptor found on cytotoxic CD8 plus T cells, and it's also found on other cells as well. And this CD28, it binds to the B7 molecules that are on the antigen-presenting cells, such as dendritic cells, yeah? And this binding is crucial. It's important in the normal sense, and in the normal T cells that haven't been in any way manipulated. You get this binding, and this binding occurs so that you can get full activation of your T cells. Now, as I said, the body produces ways, if you like, of preventing an overstimulated T cells, you know, and one of that ways is this CTLA4, the cytotoxic T lymphocyte associated proteins 4, which compete with CD28 to bind with B7 molecules and antigen presenting cells, and therefore it reduces the activation in the early immune responses, yeah? And in cancer, tumor exploit this suppression, and it exploits the fact that this, you know, it's a natural thing that occurs, but it exploits this initial priming of the T cells. So by utilizing these checkpoint inhibitors, these anti-CTLA4, it blocks this CTLA4, yeah, and therefore allows full T cell activation and promotes a stronger immune response against the tumors. I think it's been explored, from what I've been able to see, it's been explored with the use of cell-based medicinal products such as CAR T and T cell receptors in an experimental and clinical settings, yeah. However, there are there are not dangers but caveats with regards to overactivation and you know the potential for side effects. So, you know, we have to has this cautionary approach to the use of combining these checkpoint inhibitors with other medicinal products. I guess it depends upon the aggressive state and the state of or the stage of the cancer, what kind of approach medics would take, whether or not they would be prepared to take a risk with regards to the side effects, if they can manage those side effects by giving anti-inflammatory drugs, or if they can use another approach. So because I started looking at all of these checkpoint molecules, there's a lot of them, and I just thought it'd be easier to put it in a table. So I've talked about PD1, PDL1 and CTLA4. I think I talked about lymphocyte activation gene 3. So I talked about that. TIGIT, T cell immunoreceptors with Ig and It TIM domains, this competes with a molecule called CD226. So this molecule, it activates T cell and natural killer cells. CD226 combines with CD155. I think I talked about this, so I don't need to talk about it again. But TIGIT, it competes with CD226 and therefore suppresses activation. But TIGIT inhibitors are in clinical trials, but so that's a sort of checkpoint inhibitor, potential checkpoint inhibitor. I just want to give a recap on T cells. I don't want to talk in too much detail on it. But T cells are part of the body's adaptive immune response. Yeah, so are B cells. The B cells differentiate, some of them differentiate into plasma cells that can produce antibodies, which can neutralize viruses before they infect the host. B cells malignancies are what we use genetically modified T cells to address, but just talking about recapping on T cells, there are different types of T cells. So we have this cytotoxic CD8 plus T lymphocytes. Now these are the killer cells, if you like. They recognize antigens that are presented on major histocompatibility class 1 molecules, or HLA class 1 molecules. And in humans, they recognize those antigens and they bind with the epitopes on the antigens, and the CD8 co-receptor binds to the major histocompatibility complex molecules to stabilize that binding, and then they transmit a signal, and they then go off and become activated and then can kill these cells that are either infected or cells that are carcinogen or cancerous. And then so there's cytotoxic CD8 plus T lymphocytes, and then we've got CD4 plus helper cells, and these only respond to major histocompatibility class 2 molecules. So they are not presented on normal cells, they're presented on cells like, for example, antigen presenting cells, basically. And then we've got regulatory T cells, and they help to suppress excessive immune response, and to maintain that immune balance so that we don't get overactivity and we don't get the T cells attacking healthy cells. And then we have memory T cells, which are important for providing long-term immunity by responding quickly to previously encountered antigens. And again, this is just a recap that T cells develop in the thymus, where they mature into naive T cells, and then these naive T cells are then activated by binding with major histocompatibility complex antigens presented to them. And once they're activated by antigen presenting cells, the cytotoxic CD8 plus cells can then enter circulation and then kill either infected or cancerous cells. And the CD4 plus helper T cells, they just coordinate this immune response. So that's just a top level recap, if you like, on T cells and what they are. But obviously there's a lot more to this. It's just the top level. So I just want to talk about kind of moving and how I moved right from antibodies towards T cells. I was looking at the binding of the FC, the constant region of the antibody, to CD64. This is the FC gamma receptor 1, and then this can then become engulfed, if you like, via endocytosis and processed via the endosomes or in the endosomes and lysosomes. and then the processed peptides are then loaded onto these major histocompatibility complex molecules that are in the antigen presenting cells. And then they're presented to the T cells by these antigen presenting cells. Now, I was looking at dendritic cells, and they can, I think it's called cross presentation, where they can present to class 2 presentation for exogenous antigens, but they can also do class 1 endogenous antigens. So major histocompatibility class 2 presents exogenous antigens to CD4 plus helper cells, helper T cells. And you remember that because whatever, it's always got to add up to 8, and if it's class 2, 2 times 4 makes 8. So it always adds up to 8, whereas class 1 is CD4 8 times 1 is 8. Yeah, so that's how I remember it. So your major histocompatibility class 1 presents exogenous antigens to CD4 plus helper cells, and therefore that coordinates the immune response by activating other immune cells. But your major histocompatibility class 1 that presents endogenous intracellular antigens to the CD8 cytotoxic T cells, which can then directly kill that virus infected or cancerous cells. So this antigen presentation leads to T cell activation. So you go from a naive T cell to an activated T cell, and it triggers an immune response. Yeah, and monoclonal antibodies can enhance the T cell effectiveness and improve your clinical outcomes. Yeah, but so your T cells are become more better, if you like, and more at killing the infected cells, or it can be better at killing the cancerous cells. So as I said, antigen presenting cells are macrophages and dendritic cells, and they internalize the antibody opsonized antigens and then process them and display them to the, well, they display the antigenic peptides on the major histocompatibility complex molecules, or the HLA molecules in humans, basically, for activation. Yeah. Right, so I think I was getting quite carried away with myself here, but but it doesn't show everything. So this doesn't just happen in the vacuum. This is really the activation of naive cells, yeah, because before the T cells can actually, these are the cytotoxic CD8 plus cytotoxic T cell, T lymphocytes. Before they can actually go out and kill cells, they have to be activated. So these naive cells in the thymus gland, they are activated by the antigen presenting cells. So in this particular case, we have the dendritic cells, and it can engulf the infected or cancerous cells via phagocytosis, and then it processes their antigens, and then these proteins that are within the cells, the infected cells, they're degraded by proteasomes, enzymes basically, into peptides, and then the peptides are transported to the endoplasmic reticulum via the transporter associated antigen processing route, and then the peptides bind to the major histocompatibility class 1 in the endoplasmic reticulum, and then this particular complex, the class 1 major histocompatibility class 1 complex with the antigen, is transported to the cell surface for presentation to the CD8 plus cytotoxic T lymphocytes. So then these T cell receptors that are on the CD8 plus molecules, and with the CD8 co-receptor, they bind to the cell. It binds to the cell to the major histocompatibility class 1 peptide complex on the antigen presenting cells, and then you have this other action, or if you like, stimulation that has to occur, and that's a CD28 on the T cells. They have to bind to the B7 molecules, which is the CD80 or CD86, which are on the antigen presenting cells, and this provides a second stimulation or second activation, and it's called the co-stimulatory signal, and it's required for full activation. And once you get this full activation, the activated the CD8 cytotoxic T lymphocyte cells can then proliferate, and they can exert their effector function. So then these activated cytotoxic CD8 plus T cells then enter the circulation, and then when they encounter the infected or the cancerous cells, they induce cell death via perforin and granzyme B, and it's a controlled cell death. It's not sort of a cells bursting and causing more problems. It's basically apoptosis. And additionally, there's this other potential route, which is the Fas ligand, Fas-L on the T cells, and the Fas CD95 on the infected cells or cancerous cells, and they can trigger apoptosis via extrinsic pathways. So that's kind of what happens in the case of the activation of the T cells from naive cells to the activated cells. So what happens when these cells are activated? They undergo functional and phenotypical differentiation. So they're different, and they have a number of changes that make them different. They start to express different proteins that are not present on the naive cells, and they also secrete various cytokines. So the effector T cells that are formed, these are short-lived, but they actively fight infection or cancer. And then they produce memory cells, which are longer-lived, but they they're there to produce a quicker response upon re-exposure. And then they have these regulatory T cells that suppress excessive immune responses. So I started to try to put together a pictorial representation of the activated T cells, and then I realized that there's too much to put in the one sort of picture of this cytotoxic, the CD8 plus cytotoxic T lymphocytes. So I then decided to put it in a table format, but you can see that the naive cell is differentiated and is different and has different properties and functions compared to the activated T cells. So here is a table of what happens when these cells become activated. You get the expression of surface proteins, you get the expression of cytokines, and something else which I can't for the life of me remember. But you get changes. Let's look at the next one. Yeah, metabolic and signaling changes. And this is what happens. So it's a lot to put down in a pictorial representation. And even this isn't the full explanation, because we also have the CD226 that interfaces with the CD155. So I put it in tabulated form, and you can have a look at that in your own time. Right, I want to talk about genetically modified T cells, because the T cells do perform a very important task of defending the body against infected and cancerous cells, but they can be exploited by genetic manipulation to specifically target cancer cells. And this exploitation of the T cells results in two types of therapies. There's the chimeric antigen receptor T cell therapy, which have already activated T cells. So they do not require major histocompatibility class 1 molecules to bind and destroy the cancerous cells. Well, they're already activated and they don't require these major histocompatibility class 1 molecules because they are designed to be targeted towards specific antigens. And then we have these T cell receptor therapies, which do require major histocompatibility class 1 molecules, or HLA class 1 molecules, to bind and destroy cancerous and infected cells. Well, no, cancerous cells, because this is what it's designed for. So they are applicable to intracellular proteins that are derived from the antigens. So I borrowed this from I can't remember where I got it from, but I'll put a reference in the description below. And this is just showing a pictorial representation of CAR T cell receptor, or CAR T cell, and for CAR T cell therapy. So basically it consists of 3 domains. It consists of the antigen binding domain, which is it's really been taken from, or extracted from, or grafted, if you like, from an antibody, the variable regions of the antibodies. And then you have you have a hinge, and then it's attached to a transmembrane domain. So that's another region. And then the third region is the signal domain, and that contains the T cell activation domain, which is the CD3 zeta, and then you have this co-stimulatory domain that's already built into it. So remember before, the co-stimulation occurred through the CD28. In this CAR T cell therapy, you don't need to have that. It's already built into, or it's already designed into the molecule. So you have this already activated T cells. And then this was me putting together my very awkward, if you like, T cell receptor T cell therapy, where here the T cell has, is designed to bind to the antigen that is presented on the major histocompatibility complex class 1, or the HLA class 1 mutated cell. So you have this tumor cell where it contains the mutation, and then you have the neoantigen that binds to the T cell receptor. It binds to the T cell receptor. So it's different in the way that it is activated, and it's different in the way that it works, in the sense that T cell receptors are designed against, or targeted against, intracellular antigens, whereas CAR T are targeted against known antigens like the CD19 and BCMA. So BCMA, CD19 is a molecule that's found on B cells, mainly most B cells, and so where we have B cell malignancy, you can just simply target those molecules and kill those, target those cells and kill those cells through the CD19. And then the BCMA, this is the B cell mature antigen that's present on mutated cells that can be targeted. So CAR T has a different approach. It targets specific molecules, if you like, or specific surface molecules, whereas TCR T cell receptors, they target antigens that has gone through via this major histocompatibility class 1 route. So what happens, this is just a really top level overview. And what happens is you have, what's the process, if you like, the treatment process, is you have this leukapheresis, where it's a procedure where you draw out blood from the patient, and the machine separates out the white blood cells that contains the T cells from the rest of the blood. The rest of the blood is returned into the body. So then the cells are then what's the word I'm looking for? Manipulated. It's manipulated by transfecting, if you like, via this viral vector, this lentivirus vector, the transgene that can produce the CAR-T, or that can produce the receptor of interest. The cells are then isolated, and then a particular cell at best is expanded and purified. These have to be done. The transfection has to be done to good manufacturing practice. Cell expansion has to be done to good manufacturing practice. Leukapheresis has to be done to current good tissue practice. And then once you've got enough of the cells that have been expanded and purified to registered conditions, to an approved specification, these are then transfused back to the patient. If it's transfused back to the same patient, it's an autologous medicinal product. And what you really want to, when you're designing these products, is you want to get enhanced clinical performance. You want to think about what could go wrong and you want to reduce those safety events, and think about how you can target your product in order to best perform. And this is based upon experience of using, say for example, CAR-T. There's been various updates, if you like. Previously the co-stimulatory was CD28, and then that got updated, if you like, to 4-1BB because of the fact that the CD28 was, had a lot more immune effects, or a lot more side effects. And one of the side effects is cytokine release syndrome, CRS. And cytokine release syndrome is a severe immune reaction that's caused by your rapid activation of your T cells. And it can lead to high fever, low blood pressure, but it can also lead to organ damage, and so that can be controlled via anti-inflammatory drugs. So it depends, and we're aware of it, but the use of a different kind of co-stimulatory was found to be better. So this 4-1BB, the survival and proliferations of the T cells were found to be better. And then the other thing is neurotoxicity. So neurotoxicity is where you get damage, neurological damage of your nerves and sometimes of the brain. And you can have, patients can experience neurological side effects, which can include confusion, seizure, or encephalopathy. So T cells, CAR-T cell uses, as I said, it uses a chimeric antigen receptor. And this is really, I should have said this in the previous slide, but it's really an engineered protein, and it combines the antibody-derived binding domain with the T cell signaling domain. So it recognizes surface antigens on tumor without the need for major histocompatibility complex presentation. And the examples are CD19 targeted CAR-T cell therapies for B cell malignancies. Kymriah and Yescarta are examples. And then you've got your T cell receptor therapies, T cell therapies, where, as I said, it uses modified T cells to recognize antigens presented in major histocompatibility class 1 molecules. And it's really for intracellular peptides that are presented as complexes. Researchers are exploring combining immune checkpoint inhibitors with monoclonal antibodies with cell-based therapies. However, as I said, the problems with regards to the side effects have to be considered. If you are really have activated T cells, then you're increasing the chances of cytokine release or immune response, what's it called? I can't remember the word. Immune reactions, severe immune reactions. So I think I've already talked about this, where CAR T cells use a chimeric antigen receptor. It's an engineered protein that combines an antibody-derived binding domain with the T cell signaling domain. And yeah, I've already talked about this, I think. I think the only thing that I didn't say really is that CAR T cells are used mainly for blood cancers, whereas TCR therapy is used for solid tumours, where it can target intracellular tumour associated antigens. And here, I just thought I'd put contrast and compare the features with regards to CAR T cell therapies and TCR T cell therapies. And it's just I guess a summary of what we've already discussed, really. The only thing that I wanted to talk about was the restrictions. For TCR T cell therapy, it's limited to patients with compatible HLA alleles. So TCR, it does have a broader range of tumours, yeah, so it does include mutated oncogenes, but it's limited by this major histocompatibility dependence and the potential for off-target effects. So the reason for this is that the tumor antigen peptides bind strongly to one specific HLA allele or one molecule, but not necessarily to the others. And so because of that, the TCR T therapies are restricted to certain HLA types. And if you're not, you run the risk of rejection, if you use the wrong type of, if the patient is not compatible with the treatment. Oh yeah, this was just my thoughts with regards to the problems associated in real life, and that is that patients who, especially those who've got advanced cancers or aggressive cancers, the number of functional T cells may be compromised. And you're going to have problems effectively harvesting enough T cells for the therapy, and you have to consider the quality of the T cells after modification and expansion, because obviously the quality of your finished product very much depends upon the quality of your starting materials. So the functional quality of these T cells can be impaired because of exhaustion, because of this immunosuppressive environment, and you will get reduced numbers. I guess the strategies to address this, due to the, aggressive and advanced cancers, it could be like preconditioning of your T cells. If you expose your T cells to cytokine or growth factors to boost their activity and restore some of the functionality, you could also select the higher quality T cells. And then you need to consider your genetic engineering and your activation process, and you need to optimize your ex vivo expansion and culture conditions. Yeah. Yeah. So that's the end of my presentation. I hope that you enjoyed it. If you did, please consider giving this a like or subscribing. Thank you so much. Bye-bye.